Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture number 30, where I will discuss about the model for biosynthesis and secretion of monoterpene indole alkaloids involving multicellular, multiple subcellular compartments. So, in the last class, I have briefly discussed about the cellular localization of both the enzymes and intermediates in which are involved in the monoterpene indole alkaloid production, such as vindoline and catharanthine, as well as serpentine, azmalicin and so on, and these two joins together by the action of peroxidase and forms the vinblastine. And also I have shown a cross section of the leaf where different cells, where I have shown the localization of different cells such as latissifer cell, such as idioblast cells. So, where these indole alkaloids accumulate, or in other words, the late steps of indole alkaloids are operating there, whereas the most of the steps of indole alkaloids are accumulating in the epidermal region. And also I have mentioned that this latissifer and idioblasts, these are the cells which accumulate latex, and the last monoterpene indole alkaloids which are apparently more toxic. So, that basically stored in the latex, so that it will not hamper the normal cells of Cathanthus roseus cells and leaf cells. So, with this, what I am going to cover? So, I am going to see cover again the compartmentation of terpenoid indole alkaloids in Cathanthus roseus. So, I will draw a line diagram, and here particularly I will emphasize the role of epidermal cell in modulating the pathway. And once I finish that, then I will go to the cell specific localization of terpenoid indole alkaloids, that is linked with this, and how these indole alkaloids are basically transported from one type of cell to another. And then finally, I will speak on a specific transporter which was characterized few years back. So, that plays an important role in transporting one of the central monoterpene indole alkaloid intermediates to vacuole. So, with this, now let us go to the board and I will draw the diagram. So, this is basically biosynthesis of monoterpene indole alkaloids involves multiple subcellular compartment and cell types. So, this is more or less the continuation of the previous class, that is class number 29. Okay. So, let us draw quickly draw a schematic plant diagram of a plant cell. So, here this one is the mesophyll cell, which I put it in green color, and the other one which I put now in light green color, that will denote the leaf epidermal cell. So, basically this is leaf epidermal cell and this one is the mesophyll cell, and the other region what is outside, that is basically the cell surface. Okay. So, and inside this mesophyll I am also going to show these two specialized structure. One is this idioblast or latissifer, and also you have the one specific cell exists which is called phloem parenchyma. And then inside in one of the cells it contains the plastid. This is chloroplast. Okay. Now, very briefly I project this. So, for example, the MAP pathway is operating inside the plastid and that ultimately leads to the formation of geraniol, which comes out and eventually it makes 10 hydroxygeraniol, and then this will move out from this mesophyll and enters into the leaf epidermal cell, where this will be converted into loganic acid. So, this is a multiple step process, and then it makes secologanin, and that joins with tryptamine, which comes from tryptophan, and that makes strictocidin, and this strictocidin subsequently converted through common intermediates into different products. So, one particular route makes tabarsonin, and this tabarsonin finally makes 16 methoxy tabarsonin, and that moves inside the mesophyll cell again, and there this is again chloroplast, and (16-methoxytabersonin) enters into the chloroplast, and by the action of NMT it forms this, finally the diacetylvindolin, and from here it moves to the ureoblast and latissifer cells. So, here there are involvement of multiple enzymes, okay. For example, DAT and D4H is first and then DAT, and that leads to the formation of vindolin. So, the enzyme NMT is here, this is STR, this is TDC, this is SLS, and so on. Okay. Then T16H2, then T16OMT, and so on. Similarly, from streptozidin, so another branch contributes towards the formation of catharanthine. So, this catharanthine, I will put it in a different color, CATHARANTHINE. So, these catharanthine mostly, this is the most important part, mostly it moves out the cell and retained in the cell surface. And only a portion of catharanthine can be transported from here through here to here. So, that eventually joins and make the dimeric indole alkaloids. So, interesting part is this catharanthine is getting transported out from the cell into the cell surface, and here I will talk about one specific transporter that plays an important role in transporting this out, which is, these are the transporter molecules here. So, these are CRTP-T2, this is the transporter. So, catharanthine accumulates in the leaf surface, and along with this in the leaf surface you will find lot of wax. Very long chain fatty acid plus this epicuticular wax, these are accumulates there. Now, question is that what role catharanthine plays, and second point is that although sufficient catharanthines are produced in the cell, but most of the catharanthine are transported in the cell surface, and only limited catharanthine are available for carrying out the reaction leading to the formation of vinblastine by joining with the vindoline, and that is one of the reason that why the yield of vinblastine is so less. So, the next question is that why catharanthine accumulates in high amount, because catharanthine basically protects the leaf from fungal attack, fungal infection. Along with that, there are certain minor insect attack which can also be prevented by the catharanthine. So, catharanthine is basically a very important defense compound. Okay. So, the one way to isolate catharanthine is basically one can collect the leaf and then the leaf to be immersed in chloroform for around 10 minutes, and then what will happen, all the catharanthine will be transferred from the leaf surface into the chloroform, and later this can be purified and collected. So, that is one of the way which catharanthine can be collected, and that catharanthine can be used for doing the joining, right. Okay. So, and another important point, what is what I must say here, that sometime wounding happens in the cell, and then when wounding happens, that basically stimulate the vinblastine formation. So, wounding stimulates bis indole alkaloids formation. Bis indole alkaloids means this dimers. So, this is all about the involvement of leaf epidermal cell and the mesophyll cell. So, the mesophyll tissue, rather I will not write the mesophyll cell, I will write here the mesophyll tissue, that is a better word, because it consists of 3 different cells. One is this idioblast laticifera, one is the normal mesophyll palisade parenchyma, and another one is basically the phloem parenchyma. So, this is the palisade parenchyma. Okay. Once this is clear, and I have also mentioned that the idioblast contains the enzymes like D4H and DAT, and leaf epidermis contains most of the enzymes of this pathway. Okay. So, now with this I move into the next point of our discussion. Let us go to the slide. So, cell specific location of terpenoid and indole alkaloids in Catharanthus roseus leaf tissue. So, here I will now try to put my points. So, this is cell specific localization of terpenoid indole alkaloid in Catharanthus roseus leaf tissue. So, here one important point to mention, that the vindoline particularly which accumulates in the aerial part, that is the above ground parts which contain the green tissue, right. So, and you will always find the vincristine, vinblastine only in the green tissue. Now, the content of the alkaloids, what you will find in the stem and what you will find in the leaf, it differs, and what scientists they have found that the leaf tissue contains more alkaloid and as the leaf tissue matures, the level of accumulation is more. So, from there scientists they try to work out that whether there is any relationship between this leaf development and maturation and the alkaloid synthesis. So, this will be clear from a bit of drawing. So, like this is the area of leaf primordium, and here the red indicates the vascular tissue, and from here the leaf originates. Scientists, how they have localized these alkaloids, that is very interesting. So, they have used the mass spectrometry, that is a technique called imaging mass spectrometry along with single cell mass spectrometry. So, using these techniques, scientists were able to locate even the density of the alkaloids that accumulated in the leaf tissue. For example, say the alkaloids which accumulated in this area, it is shown in this turkish blue, okay, and as the leaf expanded, the level of alkaloid accumulation enhances. So, this has been demonstrated using these techniques, and this is basically showing the expansion of leaves. So, there are two particular type of cell, one is the leaf primordium cell and one is the elongating cell. So, if I make a drawing, so the bottom one what I am drawing, this is basically the idioblast cell, and the top one what I am drawing. So, this is basically the epidermal cell. So, better I use the same color here. So, this denotes the and this denotes the elongating cell. So, I will not draw the pathway in the primordium, only what I would like to tell here, that here the level of vindolin is very low, whereas here the level of vindolin is high. Along with that, vindorocin is also produced, which is basically dimethoxyvindoline. And what I have shown in the previous class, that this the top one is okay. One more thing I have to mention here, that this is the epidermal cell, and the bottom one is the idioblast or latissifer cell. So, here the from tabarsonin, for example, so that means this acetoxyvindoline, and the enzymes responsible for these reactions are this T16H2, then T16OMT, then T3O, T3R, then NMT, and so on. And then finally, desoxyvindoline, it enters from here into the latissifer or idioblast cell, and then that will be acetoxyvindoline. Now, my point of emphasizing this to you is this, that how this enters here. So, how this enters here, the question is that whether there is any existence of a transporter. That means, to get these things, okay, I use that means, to get these things out from the epidermal cell and to enter inside the latissifer cell, this requires specific transporters. Which are not yet characterized, and so this is very important. And then subsequently, this by the action of which I have said many times, that D4H and D8 it makes. Similarly, through another pathway from tabarsonin, this indole is produced. So, the point here is this, how these metabolites are transporting out from the epidermal cell and entering inside the idioblast cell. So, that is not known, and that needs to be discovered. So, that plays an important role further for this metabolic engineering application. So, and this one is and the leaf primordium, it has the similar drawing. So, I am not drawn, but what important is this, the vindoline content is much more here, whereas vindoline content is less here. So, that is also indicated from this drawing, and this one this paper was discussed by Yamamoto et al. In this journal publication New Phyto Elo GIST 2019 volume 224 to 848 to 859. So, here in this paper they have shown very interesting mass spectrometry images. So, when I will make an overview of total indole alkaloids, so I will use the slide and there I will show you those images. So, let us go to the last part of this class. So, this is also in connection with this one, which is the NPF transporter. So, the last part is basically I will very briefly talk about a transporter. This transporter is called NPF transporter. This transporter was shown to exports a central monoterpene indole alkaloid intermediate from the vacuole. So, and this transporter has been characterized. So, let me quickly draw a diagram to explain this. So, this is basically the diagram of the cell. So, here I will bring the issue of this early steps of indole alkaloid, that is secologanin and tryptamine, and that makes strictocidin. So, this is the cell, and here with this color I mentioned that this is basically the vacuole. So, this is vacuole, VACUOLE, and the remaining portion is the cytosol or cytoplasm. Okay. So, secologanin and tryptamine, they enters into the vacuole, and this must be mediated by some putative transporter, and these two once enter into the vacuole, these two joins together and form the molecule strictocidin. Okay. This strictocidin, once it is produced, so these strictocidins will be transported out from the vacuole into the cytosol, where this strictocidin will be undergoing detachment of the sucrose, leading to the formation of strictocidin glycone, and which will be subsequently utilized for running multiple pathways leading to terpene indole alkaloids. So, these all we know. We have said that this is SGD, this is STR, but what is to be discussed now is this one. There is a transporter here which transports this out, and this is the CR NPF 2.9. So, this transporter has been characterized, and which plays important role in transporting the strictocidin out from the vacuole into the cytosol, so that strictocidin subsequently be deglucosylated by the action of strictocidin glucosidase. So, this is very important. So, and so far only 2 transporters were characterized in this Catharanthus system. One was ABC transporter, an ABC transporter, and that exports catharanthin into cell surface, and the second one is basically a proton driven antiporter which transports vindolin and let me write vindolin and catharanthin into the vacuole for subsequent conversion. Now, how they have checked the function, that is very interesting. When they have characterized this transporter, that is when they have the gene. So, they have silenced this. So, they have made the silencing, that means silencing of rather I will write implantar silencing of CR NPF 2.9, and then what will happen. So, that will be very clear if I draw 2 simple chromatogram to explain. So, one for example is your control. So, where this is basically the LC MS chromatogram, LCMS ion chromatogram, sorry, LCMS chromatogram showing strictocidin. So, this is the control. So, the control means that this is Higgs with empty vector, and the one which is the real one. So, that means the silenced one. So, that will show so this is now if I highlight with the red one, then it will be clear. So, this peak is basically for the strictocidin, which is present in Higgs silenced plant, whereas in the wild type plant or the control plant, strictocidin is not present, because strictocidin will immediately convert it into strictocidin aglycone. So, this is the way to characterize once you have made the function disrupted. So, strictocidin cannot be transported out, and therefore, if extract is made and run in LCMS, strictocidin can be detected. So, this is a very unique work, and it is a very significant discovery. That is why this was published by Pine et al. And the group leader is of course Sarah E. O'Connor, and that time she was in John Ennis Center, UK, and this paper published in the famous journal Nature Plants in the year of 2017, volume 3, article number 16208. So, with this I have given you more or less the overview of the compartmentation and the role of transporter, which plays important role in transporting these metabolites. So, with this I end this class. Thank you very much.